Good day, students. In today's class, we want to look at excitation, contraction, coupling in cardiac and smooth muscle. Would have previously studied excitation, contraction, coupling in skeletal muscle. And what we want to do in this class is look at the unique features of cardiac and smooth muscle and see in what circumstances is excitation, contraction, coupling the same, and in what circumstances is excitation, contraction, coupling different. In other words, we can say we're going to compare and contrast excitation, contraction, coupling in skeletal, cardiac, and smooth muscle. As we begin, let me just remind you that the term excitation, contraction, coupling refers to the process by which a nervous impulse brings about contraction of muscle tissue. So as usual, I'll just put this list of learning objectives for you. In order to understand excitation, contraction, coupling in cardiac and smooth muscle, we'll have to review the histology and just talk about the different places where these types of muscles are found in the body. Then we're going to talk about the action potentials found in cardiac muscle. And then finally, we're going to talk about specialized processes in the different muscles that allow them to perform their unique functions. So let us begin with cardiac muscle, and let's just review some of the unique features or the functional anatomy of cardiac muscle tissue. Remember, we would have said that skeletal muscle tissue has very long fibers or very long cells. Well, the cardiac muscle cells are also long, but perhaps not as long, and they often tend to be Y-shaped or bifurcated, and they can have one nucleus or two nuclei. And so this immediately gives you one difference between skeletal muscle and cardiac muscle. Whereas skeletal muscle is multinucleated, cardiac muscle has at most only two nuclei. They tend to be shorter and wider than skeletal muscle cells. However, just like skeletal muscle cells, they have a regular arrangement of sarcomeres with thick and thin filaments, and as such, they appear striated. So if you look at cardiac muscle, it will have the same banding pattern that you'd see of skeletal muscle under a microscope. Cardiac muscle has a large number of mitochondria, which talks about the role that cardiac muscle has to play in constantly beating and the fact that that energy has to be supplied by a continuous oxygen delivery to the tissue. And cardiac muscle has T-tubules that tend to be larger, up to 5 times in diameter when compared to skeletal muscle tissue. And we'll see the importance of that in a little while as it relates to excitation, contraction, coupling. The other major difference that you'll notice between cardiac muscle cells and skeletal muscle cells is cardiac muscle cells have a series of specialized gap junctions, which are termed intercalated discs. And these allow the cell membranes of the cardiac muscle cell to be fused along their longitudinal axis. Now we know that gap junctions, or intercalated discs in this case, allow a rapid transfer of action potentials and signaling from one cell to another. The net result is it allows the cardiac muscle to function as two syncytium. A syncytium is a connection of cells that can function as one. So you have an atrial syncytium and a ventricular syncytium. And the advantage of this is this allows almost synchronous contraction of the entire atria and the entire ventricles at the same time, allowing the blood in the heart to be forced out, as opposed to a slowly developing contraction as the signal traveled around or along the muscle tissue. So that just gives a brief recap of some of the histological features of cardiac muscle. Now let's remind ourselves of the cardiac muscle action potential, because this action potential has one feature that's very, very important for cardiac muscle function. And you can see in this diagram a typical cardiac muscle action potential. And of note is this segment labeled 2 here, which we call the plateau phase. This is a segment that you don't see in the action potential of a nerve. The action potential of a nerve is very rapid. It's over within 4 or 5 milliseconds. But the cardiac muscle action potential is much longer. It can last for up to 200 milliseconds. And that's because of this plateau phase. And the plateau phase is generated by the opening of specialized voltage-gated calcium channels. And these calcium channels open a couple milliseconds after the opening of the voltage-gated sodium channels. And what they allow is an influx of calcium into the cell. And this prolongs the depolarization of the action potential until the calcium channels close and the sodium channels open and the membrane begins to repolarize. Now, this calcium channel that allows calcium into the muscle cell allows some very unique and important features of muscle tissue. The first thing I want to mention is this functioning prevents heart muscle going into a state of tetany. Now, tetany occurs if a second action potential arrives at the muscle tissue while that muscle tissue is still undergoing a state of contraction, and it produces more contraction in the muscle tissue. In skeletal muscle, that leads to increased force of contraction. But if pushed to the limit, it can result in skeletal muscle undergoing a state of sustained and prolonged contraction, which we would describe as tetany. We do not want that to occur in cardiac muscle because if the ventricles and the atria were to go into a state of prolonged contraction, they would not relax and allow refilling, and the pump would essentially shut down. Remember, cardiac muscle is a pump. The heart is a pump. And if the atria and the ventricles go into sustained contraction, then they can't fill, they can't refill with blood, and the pump would fail essentially. So the fact that the duration of the action potential and the amount of time free calcium remains within the sarcoplasm are nearly the same basically prevents another action potential firing before the contraction relaxation of cardiac muscle tissue is complete. So you can add more calcium to the muscle cell, but it's not going to trigger another contraction because there's already a high level of calcium inside of that muscle cell. And that prolonged period of time during the plateau phase ensures that cardiac muscle does not go into tetany, which is very, very important. Now, this also explains to us another major difference between cardiac muscle cells and skeletal muscle cells. Essentially, the process of excitation, contraction, coupling in cardiac muscle is almost exactly the same as in skeletal muscle. But the one real unique difference is the source of the calcium that triggers the process. Now, in skeletal muscle, in cardiac muscle, and in smooth muscle, the trigger for contraction is a rise in intracellular calcium. We call this the calcium transient. In skeletal muscle, we saw that calcium originates from the sarcoplasmic reticulum. And that sarcoplasmic reticulum gets activated when there's a depolarization spreading down the T tubules, which gets transferred from the T tubules to the sarcoplasmic reticulum. And we looked at the triad and we looked at the interaction between the dihydropyridine receptor and the ryanodine receptor and how that allows the transfer of the depolarization from the cell membrane to the sarcoplasmic reticulum. Well, in cardiac muscle, the initial source of calcium is different. The initial source of calcium that triggers the first part of contraction is the calcium that enters the muscle cell from the extracellular fluid, as we just saw in the cardiac muscle action potential. When that calcium comes into the muscle cell through the cell membrane, it interacts with receptors, the same receptors on the sarcoplasmic reticulum, which triggers a further release of calcium from the SR. And so we call this process calcium-induced calcium release. In skeletal muscle, the calcium release is controlled or triggered by depolarization. In cardiac muscle, the calcium release is triggered by the influx of calcium through the calcium channels on the cell membrane. Calcium-induced calcium release. And now we can see the importance of having these larger T tubules, because these larger T tubules allow more extracellular fluid to come close to the cell membrane, allowing more calcium to be present close to their channels. And this then allows the calcium to flood in once that cardiac muscle action potential originates. So let me just repeat. A major difference between cardiac muscle and skeletal muscle is in cardiac muscle, the initial trigger for contraction and the release of calcium comes from calcium flooding into the cardiac muscle cell through calcium channels, found on the cell membrane. Well, the other major difference is how do you remove the calcium? And in cardiac muscle, we have special sodium calcium exchangers that are also found on the cell membrane. Now, these sodium calcium exchangers exchange one calcium for three sodium. So these pumps allow one calcium to exit the cardiac muscle cell, and in exchange, they bring three sodium into the cardiac muscle cell. And the sodium calcium exchanger uses the natural electrochemical gradient generated by the fact that there's more sodium on the outside of the cell than is on the inside of the cell. So as sodium flows down its concentration gradient through the calcium exchanger, the energy released is coupled and used to take one calcium out of the muscle cell into the extracellular fluid. And this is how we remove calcium. We also have a few pumps like this on the SR, but they have a much smaller function. Now, this sodium calcium exchanger then is very sensitive to the sodium concentration gradient. If the sodium concentration gradient between the extracellular fluid and the intracellular fluid changes, then it can actually even reverse the activity of this pump, where the pump can start pumping sodium out and bringing calcium in. And we can use this in clinical situations. Particularly, we can use it to change the amount of calcium in the muscle cell and increase or decrease the strength of contraction. And one drug which you've previously studied that does this is a group of drugs known as cardiac glycosides or digitalis, of which an example is digoxin. Now, what digoxin does is it inhibits the sodium-potassium ATPase pump. Now, remember what the sodium-potassium ATPase pump does. That pump pumps sodium out and brings potassium in. So if that pump is not working, it leads to an accumulation of sodium inside of the cardiac muscle cell. And it basically alters the gradient of sodium across the cell membrane and therefore diminishes the function of the sodium-calcium exchanger. The end result is less calcium is pumped out of the cardiac muscle cell. More calcium remains in the cardiac muscle cell, leading to a greater strength of contraction. And so we use digoxin and digitalis and these cardiac glycosides when the heart is failing and we want to increase the strength of contraction of the heart by keeping calcium in the muscle cell and pushing more blood out of the heart. If for some reason we wanted to reduce the strength of contraction of the heart, maybe in cases of hypotension, we can actually use calcium channel blockers. These will stop calcium getting into the heart, reduce the strength of contraction of the heart, and they can bring down blood pressure. And in some cases, this is what is done. So you're beginning to understand some of the physiology of how if we regulate the amount of calcium inside of the cardiac muscle tissue, it changes the strength of contraction. And basically, that's how strength of contraction is regulated in heart muscle tissue. Okay, so we've looked at cardiac muscle and we've seen that its systems are very similar for excitation contraction coupling as occurs for skeletal muscle. But let's talk a little bit now about smooth muscle. And what we're going to see is that smooth muscle actually has very, very different systems. Now, we'll just remind ourselves that many organs, almost all organs of our body have smooth muscle tissue. Your digestive tract, your respiratory airways, your blood vessels, your stomach, your bladder. And these structures often require the smooth muscle to be active over long periods of time. And once muscle is active, it requires ATP. And so one of the things we're going to see is that smooth muscle is adapted to utilizing the least amount of ATP over the time of contraction. The other thing you'll notice about smooth muscle is they have much smaller cells. They're spindle-shaped, and they have a single central nucleus. Smooth muscle cells are also not striated. They do not have the regular banding pattern because the actin and myosin are arranged not in a haphazard fashion, but they're not all arranged in the same axis, which talks about the fact that smooth muscle often functions to change the dimensions of vessels, and they need the actin and myosin filaments moving in different axes. So typically you find smooth muscles arranged in layers around hollow organs. And you would have known this from your study of the digestive tract, that they can be circumferentially arranged to reduce diameter, as in blood vessels or in the airways of the lungs. Or sometimes they can be longitudinally arranged to allow propulsion, as occurs in the GIT. And then finally, sometimes they can crisscross one another, and this allows organs to expand and contract, as happens in the bladder. And this crisscrossing arrangement, if you look at my hands, allows expansion without much increase in intra-organ pressure. And that's what you'd want in the bladder. The other thing we can say about the arrangement of smooth muscle is it's usually found as a mass which we call single unit smooth muscle, in which only a few of the muscle fibers are innervated, but the electrical signal spreads throughout the mass of smooth muscle, and it functions as a syncytium, much like what we saw in heart muscle. In some other parts of the body, you do have what we call multi-unit smooth muscle, where almost individual cells or small groups of cells receive nervous function. But this is much less common. It would occur within the smooth muscles related to the hair on your body, the erector pili muscles of the skin, and also within the iris of the eye. And this diagram here just highlights the fact that smooth muscle is often not connected to the bony skeleton. And so the arrangement of the myofilaments within the smooth muscle allow it to contract against one another and actually change the shape and dimensions of the organs with which it functions. Okay, so let's look a little bit at the ultrastructure of the smooth muscle. And what's the first thing we notice is that smooth muscle doesn't have the typical arrangement of sarcomeres that we saw in cardiac and skeletal muscles. Smooth muscle actually doesn't have Z lines or Z discs. They actually have something called dense bodies. So the analogous structure of the Z disc in the smooth muscle is the dense body, and it's formed of a protein called alpha-actinin. Connected to the dense bodies are your actin filaments. In between the actin filaments, you see your thick filaments. However, part of the cytoarchitecture of the smooth muscle cell is that dense bodies are also connected to each other via something called intermediate filaments. And this is all part of the internal architecture of a smooth muscle cell, which provides anchoring and allows contraction to take place. We call it the cytoskeleton of the cell. The thin filaments have actin and tropomyosin, but they don't have troponin. And so we know that troponin is the molecule that receives calcium. So that's one of the things we have to consider. How does contraction occur if there's no troponin? And finally, you find that there are a lot more thin filaments in smooth muscle than there are in skeletal muscle. So the actual ratio between actin and myosin is 10 or 15 to 1, whereas in skeletal muscles it will be much more like 2 to 3 to 1. Now, how does excitation-contraction coupling occur? Well, unlike in skeletal muscle, actin and myosin do not spontaneously interact. And that's because of two reasons. Tropomyosin also exists and it blocks actin and myosin spontaneously interacting. But also myosin by itself will not just interact with actin. Myosin will only interact with actin when it's phosphorylated. So here's the process. To trigger contraction, calcium must enter the cell. Once calcium enters the cell, it doesn't bind to troponin because there's no troponin. It binds to another protein called calmodulin. And you have 4 calcium molecules binding to one calmodulin molecule. Activated calmodulin now interacts with an enzyme called myosin light chain kinase, and it activates the myosin light chain kinase. So myosin light chain kinase exists in the cell, but it's in an inactive form. Activated calmodulin activates myosin light chain kinase. Once myosin light chain kinase is activated, it takes a phosphate from ATP and it puts it on the myosin head. And that attachment of the phosphate to the myosin head activates myosin to now bind with actin. And the sliding filament theory proceeds. One of the things that we have to appreciate is in smooth muscle, the ATPase activity of the myosin head occurs at a much slower rate than in skeletal and cardiac muscle. So there's a slower rate of cross-bridge cycling in smooth muscle. And this is one of the things that allows the contraction to occur for a longer period of time. Now we can see the steps here in this diagram. Calcium comes in, it binds to calmodulin. Activated calmodulin takes the inactive myosin light chain kinase and activates it. The activated myosin light chain kinase then takes a phosphate from ATP and puts it onto the light chain of the myosin molecule. And the light chain of the myosin molecule is now in a place to interact with the actin molecule. And we have the sliding filament theory occurring. The question now we ask is, how do you stop contraction? Well, when the calcium concentration falls, another enzyme gets activated called myosin phosphatase. Whereas kinases add a phosphate, phosphatases remove phosphates. And so what this phosphatase does is it takes the phosphate off the myosin head. And once the phosphate is removed, over a period of time, the myosin detaches from the actin. And you can see that in this diagram here, where the myosin phosphatase is removing the phosphate from the myosin, leading to the breaking of the cross bridge and a decrease in tension. And so you realize the process is very, very different in smooth muscle contraction, the process of excitation, contraction, coupling. Now, I also told you that the tropomyosin is blocking the actin and the myosin from interacting. And in order to remove the tropomyosin, that's done by a protein called caldesmon, which now interacts with the calcium calmodulin complex. So let's just see that in this diagram here. So caldesmon is interacting with tropomyosin here, and it's stopping the actin filament interacting with myosin. The activated calmodulin comes and interacts with the caldesmon and removes it, and in so doing removes the tropomyosin block and allows the myosin head to bind to the actin and trigger contraction. Now, one of the things that is very interesting and is unusual is that as the concentration of calcium is falling, when it reaches a kind of middle stage where it's not at the level where the phosphatases get activated, a strange thing happens to the caldesmon in that it's able to re-engage with the actin molecule and the myosin molecule at the same time and anchor them together. Remember, this occurs at intermediate calcium concentrations. And when this happens, a latch bridge forms. The smooth muscle remains in a state of contraction, but there's no cycling of the myosin heads. And so there's not much ATP being used. And this is one of the ways in which smooth muscle is able to stay under a state of sustained contraction without using much energy. Now, there's a lot of interest into understanding the latch bridges, and there's still theories that are being worked out. One idea for the latch state is also the fact that while the myosin and the actin are interacting, the phosphatase removes the phosphate, and that also allows the latch state to occur. So we do know, let's say what we do know. We do know that smooth muscle can stay in sustained states of contraction for prolonged periods of time. We do know that the cal-desmon seems to be important in forming these latch bridges, and it has to do with the level of calcium in the cell. And so again, we see the level of calcium regulating the strength of contraction. At this intermediate stage, something strange happens where the calcium re-engages the actin and the myosin and forms a latch bridge. But as the calcium continues to fall, the myosin head eventually comes off and it moves into a state of relaxation. So, once again, the strength of contraction is determined by the amount of intracellular calcium. Smooth muscle is unique because it gets signals from nerves, it can get signals from hormones, and also there can be local factors in the environment that can cause the concentration, that as their concentration changes, it affects contraction of smooth muscle. This would be examples like changes in the partial pressure of oxygen and carbon dioxide, or H plus concentrations, or lactate. All of these things can interact with smooth muscle to change the amount of calcium in the smooth muscle cell. And if you look at this diagram here, you'll then see that smooth muscles often have multiple receptors located upon their cell that can respond to different ligands, different hormones, as well as nervous signals. And so the contraction or the regulation of calcium within the smooth muscle cell is under multiple control systems, which makes it very different to what happens in skeletal muscle cells and cardiac muscle cells, which tend to be primarily under nervous regulation. So that ends our discussion of excitation contraction coupling in cardiac and smooth muscle. I hope you've been able to appreciate the differences, and that if you were asked a question to compare and contrast excitation contraction coupling in skeletal, cardiac, and smooth muscle, you would have a clear picture in your mind of what are the different things taking place in each and why they are important. So until next time, goodbye.